This is Cheyenne and Shakoya with another Psychological and Brain Sciences Department podcast. Today we are here with Dr. Rachel Smith, a behavioral cellular neuroscience professor here at Texas A&M.
What classes do you teach?
So one of the primary classes I teach is drugs and behavior, which is 336. And so I've taught that several times. We go over a lot of different drugs of abuse, how the pharmacology of them, how they affect behavior. Then we also go over several neurotransmitter systems related to how these drugs work. So that's kind of my main class is drugs and behavior. I'm also currently teaching physiological psych, which is kind of the prerequisite intro class for a lot of the upper level neuroscience classes. And so it's a much bigger class. And then I also, every couple of years, teach a graduate class on the neuroscience of addiction and anxiety.
Nice. That's awesome. So what is it like teaching graduate students versus undergraduate students?
It's very different teaching graduate students because they should have a solid foundation of the basic concepts. And at that point for graduate school, I really want the graduate students to be engaging in critical thinking. Mm-hmm. So I want them to be reading the primary literature, thinking about it, and also being sometimes skeptical of it and really evaluating it in a critical way and thinking about not always taking it at face value, because sometimes you might have two papers that contradict each other and you need to understand, is there something different between the papers? Did they interpret something wrong? So for graduate students, I really want them to be thinking critically. I would like undergraduates to think critically too, but need for neuroscience, there's so much of a foundation you kind of need to get first before we can even get to that level.
Right. So how would you describe your teaching style, I guess, like in general, with both your undergrads and your graduate students?
So for the undergraduates, I really do more PowerPoint type lectures. I try to give lots of examples, lots of pictures, just to help, because I know sometimes, just having words on the screen doesn't really help you get an idea of what we're talking about. So I try to put in different pictures and talk about things and give examples. And I also try to sometimes relate it to some kind of more advanced things that are happening in neuroscience, just to kind of make it a little bit more exciting, or relate it to some drug abuse or something, just to kind of make it a little more interesting in terms of this is why this is important. So I use a lot of PowerPoints and that type for those classes. I also try to organize my lectures in a way that it's pretty clear what the important topics are and what I want you to know, and then I try to summarize it at the end so that it's clear, this is what we just learned, and this is what I want you to take away from it. So that's for the undergraduate classes. For graduate classes, like I said, I tend to provide more primary literature sources. We read papers. There's a lot more discussion. Mm-hmm. I grade based on discussion, because in graduate school you should be ready to discuss. Right. And the students have to make presentations as well. And so I feel like that format's better. I also make them, I ask them to write critiques on papers as well. So that way they get practice with writing skills and also get how to critically think about a paper. Right. And also how to maybe in a nice way describe how something you might be skeptical about, something like, an alternative explanation might be this, as opposed to slamming somebody's paper and being mean. So I feel like that's an important skill too when you're reviewing manuscripts, which we all do in the field, is how to be able to evaluate it without coming across, as mean or something in your evaluation.
Right.
So
what would you say your philosophy of teaching is?
My philosophy of teaching, I think that I hope that I explain things in a way that students can really retain that information, and that it makes sense, and it's not something that students are just memorizing just for the exam and they forget it later. Right. I think that a lot of students that take my class end up saying, I learned a lot, but it was really good. And so that's what I want. I want students to come away thinking, I learned a lot, and hopefully they'll remember it when they take other classes. Right. So I want to just explain things and for people to understand concepts and understand things about the brain and neurons and behavior, so that it's not something that you feel like you're just cramming and trying to memorize just for this one test. And so then I also try to relate throughout the class back to things that we learned early on, just to try to help with that continuity and remember, and to help with the memory too, because the more you can relate back to something you've already learned, the more times you recall something, the more you're going to remember it. So I also try to get this try to ask the questions during lecture, which is sometimes hard in bigger classes, but just to get students to not just be sitting there listening, but to be thinking about the information I'll say. And then, and then what do we talk about when we did this, or what do we need to remember, just to kind of hopefully get students engaged and actually thinking about the material as I'm talking about it as well.
So could you tell us a little bit about your teaching experiences?
So the teaching experience I had before starting as faculty, I didn't have actually a ton of teaching experience, I think because I'm in a biomedical science field. So when I was in graduate school, I went to University of Pennsylvania, and we had to TA, be a teaching assistant for 1 semester, and that was the only requirement. So I was a teaching assistant for an intro to neuroscience class, and then I was a head teaching assistant for another semester. And I liked being the head teaching assistant because it gave me a chance to go back and try to improve some of the things that I didn't like when I was a TA. And for that class, we had a lab component where students came in for recitation, and we also did like a sheep brain dissection. And I thought that the lab manual was not helpful at all. So like when I was the head TA, I completely redesigned it, put in new pictures, tried to make the dissection make more sense so that students would get more out of that brain dissection. And then I would give guest lectures here and there as a graduate student and as a postdoc, because in biomedical sciences, you often do a postdoc before you can get a faculty job. And then when I was a postdoc, I also helped out. I did my postdoc at the Medical University of South Carolina, and they have a lot of med students. And so the med students have to take gross anatomy, and they have to learn about the brain, and the brain often is really overwhelming to med students. And so they would recruit postdocs to come in and help with the brain gross anatomy. And so we would come in and help them find the cranial nerves, do the dissections, start to tease apart the different parts of the brain and what you can see and what you can't see. So I thought that was fun too. I like anatomy.
That's awesome. Yeah. So can you kind of explain what a postdoc is, just a little bit more?
A postdoc, so that's after you finish your PhD. In the biomedical sciences, just because there's so much training and there's so many techniques that are involved, it's pretty much impossible almost to be ready after your PhD to find a faculty position. And you're competing with people that have done postdocs and have way more papers. And so people do 3 to 6 years of postdoc usually in 1 or 2 labs, just to get even more experience, get more papers out, hopefully get a grant or something during that time, so that you're really ready to have your own neuroscience lab. Yeah.
If you could teach any course you wanted, what would it be and why?
Well, I really enjoy the drugs class that I teach because I study drug addiction in my lab. I find that drugs of abuse really interesting, and the pharmacology and talking about the brain. So that is really one of my favorite classes to teach. If I could design a new course here and we had the resources, I think it would be neat to have some type of hands-on lab training course. We had something like that when I was an undergraduate, where it was a little more advanced, and you were in the classroom for 1 day a week, and then you would go into an actual lab another day of the week, and you would learn. We ran very simple behavior on animals. We actually did animal training, and we even did a very simple surgery on them. And then we, at the end of the experiment, we had to sacrifice the animals. We got the brain out. We got to process the brain. So it was really neat just to get that hands-on experience. And so if we had the resources to do that in a space where we could do that, I think that would be a really neat course, because I don't know, I like training people on how to do actual research also.
Right. Yeah. So what is it like, I guess, whenever you incorporate animals in a lab? Do you have to go through a whole other process so that way you are handling them with care and things like that?
Right, exactly. So for human studies, you have to go through IRB, and then for animal studies, we have to go through IACUC, which is the Institutional Animal Care and Use Committee.
Oh, okay.
And so every university is going to have an IACUC, and IACUC reports to some federal agencies. And anything you're going to do in animals has to be in a protocol. It has to be submitted, and IACUC includes several faculty, veterinarians, even a general layperson is on the IACUC committee. And so they have to approve any procedures you're doing, any type of drugs you're giving, anything that's stressful to the animal, how you're doing surgery. So it all has to be approved. And then they also will do annual inspections or semi-annual. I think they're coming tomorrow. They'll come through the labs, make sure things look okay, make sure we don't have expired drugs, all that type of stuff. So there is quite a bit of oversight to make sure that we're treating animals humanely, because that is important to do.
Yeah, for sure. So can you tell us a little bit more about your research interests, I guess more in depth?
Yeah, so we're interested in drug addiction. So we use animal models to study drug addiction. So we use rats, and our rats actually self-administer a drug. So that means that they go into an operant chamber and they press a lever and they get infusions of drug. So that means we have to do surgery on the animals to put in an intravenous catheter into the jugular vein. And so then there's a little port that comes out their back. And so when we put them into the chamber, we hook them up through a leash, and then that way they're just in control. They push a lever, they get infusions of cocaine, usually in my lab, and then we can study what they're doing. Excuse me. So when most animals will take drug, especially things like cocaine. So that's not really, it's kind of the highlight of their day, and it is very reinforcing and rewarding, obviously. But that's not really addiction, right? Because just taking a drug because it feels good is an addiction. And so what we've been trying to develop since I started here 4 years ago is getting into a model of addiction that looks a little bit more closely like what we see in humans, and particularly compulsive use. And so compulsive use is when you keep taking a drug despite negative consequences. So even though there's reasons to not take it, you can't stop. And so for animals, for rats, we can't have a negative consequence of like losing your job or going to jail or losing your family. So we have to introduce some other negative consequence. And so we use a brief foot shock for them. And it's not even painful. It's just something that's like a little bit aversive. They don't like it.
Right.
And so after animals have been taking drug, they usually go in for 2 hours a day. After they've been taking drug for like 4 to 6 weeks, all of a sudden we say, okay, you can keep getting cocaine, but you're also going to get a brief foot shock. And so what happens is, what's interesting is that animals respond really differently to that foot shock. So some animals are like, well, forget it. I'm going to stop this. I don't want to get foot shocked. And some animals are like, well, I'm just going to keep taking drug even though I'm getting foot shock. And so we think both these populations are really interesting, because the one kind of models addiction, and we're trying to figure out what is going on with these animals that they keep taking drug despite getting foot shock. And we also want to study the animals that are more adaptive, I would say, and decide to reduce their behavior to avoid getting foot shock, because that's what we want to model in the other animals. And so we're looking in particular at the role of habitual behaviors in this. And so habits are defined in a pretty specific way in behavioral neuroscience. So we differentiate habitual behavior from goal-directed behavior. So when I tend to think of it like when watching somebody drive a car. If I was watching you drive a car and you turned right at a stoplight, I wouldn't know if you turned right because you were doing it in a goal-directed way or a habitual way. So if you're doing it goal-directed, you may say, oh, I need to go this way today because I have to go pick up my dry cleaning. So you're thinking about the outcome of what you're doing, and that's why you're making the action. Whereas a habit would be, you turn right and you're like, oh shoot, I didn't mean to do that. It's just like I got to the light and I turned right. So that would be habitual. So when an animal is pressing a lever, we don't know if they're doing it in a goal-directed way or a habitual way in the same way. So we have to probe it and say, were they pressing the lever because they were thinking about the outcome, the cocaine, or were they pushing it because it just became habitual? And we tend to think about drug addiction as being a habit, right? Something you can't control, but that's not really been tested a lot. And people that study humans that are addicted to drugs will often say they seem very goal-directed. When they go out and look for a drug, it's like they have a whole plan. They're this plan to get money and to do this, and it's not really like a habit. And so we had to develop a way to test for whether or not animals were habitual in terms of how they're responding for drug, so that we can really tease apart, is there really a role of habits, or is that just something that we kind of think is important? And what's really the underlying mechanism that drives animals to be resistant to punishment, is what we call it. And so we're studying the behavioral mechanisms of that, as well as what's going on in the brain, what circuitry is normally responsible for when animals get punishment and they suppress their responding versus when animals are not suppressing it. So we're teasing apart what's happening in the brain as well with animals.
That's super
That's
interesting.
really cool.
That's so cool.
Tell us how your research has influenced your teaching. In what ways have you been able to bring the insights of your research to your courses at the undergraduate level?
I think it's sometimes tricky to weave some of the things that we do in lab into undergraduate teaching because it's sometimes so far removed and so detailed that it's hard to weave in. But I do try to talk about the applications of what I'm teaching students, because I think a lot of people find drugs interesting and doing animal models and doing behavior and, so I try to bring that in, those kind of, just to make it a little bit more interesting sometimes.
Mm-hmm.
And when we, for instance, talk about different animal models that we use in neuroscience, I can kind of give more firsthand experience, like about what animals do in certain situations and these different behaviors. So I'll talk about that. And I also, because I have an active research lab, then I know a lot of the, more cutting edge neuroscience techniques that are being done. And so I can kind of incorporate, well, this is what we actually do in a neuroscience lab. Because I think sometimes people have a misconception that we still do experiments the way maybe you imagine them happening in the 70s and 80s. Like, oh, we just give an animal a drug and we watch what happens. Like, we're so beyond that now.
Right.
Like I just mentioned, we're doing this compulsive responding, and it's like, it's not, we're way past just giving animals a shot of an injection and, and seeing what happens.
Right.
So I try to incorporate that in my classes. I also think it's really important to tell the undergrads and inform them that many of their faculty, maybe not all, but many of the faculty are researchers. We are here to teach, but we're not just here. The reason we are qualified to teach is because we are researchers, and we have very active research lab. This is a tier 1 research university, so it's very active, and I think that's important for them to keep in mind, that we're not just lecturers necessarily. Not all of us. We also have really active research labs, and that's what qualifies us to be here and teach.
Right. So, like, along the lines of like how active your research lab is, how do you involve like your graduate and undergraduate students in your research?
So graduate students are pretty critical for my research, because we're doing animal behavior and running animals constantly in behavioral paradigms every day. I would not be able to do my research without graduate students. So I currently have 2 graduate students, and I'm looking for more even, and they're constantly in the laboratory running animals, running the data, collecting data, analyzing the data. And so then I work really closely. We meet at least weekly, if not more, to talk about what experiments we're going to run, how to analyze the experiments, how we're going to write up papers, all that. So graduate students are pretty much absolutely critical for a laboratory like this. We also have undergraduates that work in the lab. The undergraduates, we have about maybe 5 a semester, and we ask them to stay for 2 semesters because it's so much training to even get to them to a point where they're like actually helping. So we ask for 2 semesters, and they learn to handle the animals. They have to, of course, go through all the animal training also. They learn how to handle the animals, load them into the boxes. They learn how to cut brains, how to process brains, how to mount tissue. So they're going to learn some neuroanatomy because they're working with the brain. So yeah, they come in and they do quite a bit of hands-on stuff in the lab, and so that's really helpful as well. And then a lot of times in labs like this, you will have postdocs. Like I just mentioned, I'm a postdoc. So I don't have a postdoc currently, but postdocs could come in, and it would be someone that's already has quite a bit of training and quite a bit of experience writing, but would be kind of at a little bit higher level than at that point.
Well, thank you so much for joining us today, and we will be back soon with another interesting insight into the faculty of the Psychological and Brain Sciences Department.